So it's time to start the session 2 on radiobiology and carcinogenesis of particles, and the two chairs are CIMAC at Boost and from Karolinska University and Walter Tiganelli from Heidelberg. GSI.
So, yes, I will just say, present the next speaker. So, thank you very much for until now very exciting discussion. So we change a little bit direction and going to the little bit biology. So I would like to ask Dr. Benjamin Frey, who is associate professor at Department of Radiation Oncology, University Hospital of Erlangen, Germany, to take the floor and talk about the radiation effect of immunology. Please.
So thank you very much for this invitation, and also thank you very much for this nice introduction. So unfortunately, I have to disappoint you twice. I'm neither a radiation biologist, nor I have a complete presentation about abscopal effects. But I'm a very, very trained radiation oncologist, and I worked with Udo Geipel, my boss, together at the Department of Radiation Oncology in Erlangen since now more than 20 years together. And we call it not abscopal effect because many of the immunologists, when they hear abscopal effect, they think it's a little bit of voodoo. Yeah, therefore, we call it systemic immune modulated or mediated effect. So I had a duty from CIMAC to show you some interesting things about combination of radiotherapy and immunotherapy, and they are really brother in arms. So, but I just want to introduce our group shortly. So we are radiation oncology. We are a small group at the Department of Radiation Oncology, and we have several fields where we are working in. We are working on osteoimmunology and radiation. We are working in systemic and anti-tumor immune responses. That's what I will present today mostly. We have brain tumors where we have preclinical models. And we have molecular radio-oncology where we also do experiments on radiosensitizers because we have a strong cooperation with our chemists at our university. And we have a huge unit, I will come back later to this, where we have a clinical radioimmunobiology. That means we are doing immunophenotyping of nearly all the patients at our department and at different other departments in Germany where they undergo some radiation oncology trials. So I have divided my presentation into two parts. So the first part, I want to say the challenge one, the cancer immunity cycle. So it's very easy to start an immune answer. Our body knows how to start an immune answer, and normally he also knows how to start an immune answer perhaps against cancer cells. And it begins here with the damaged cells, and then the damaged cells will be taken up by the dendritic cells. They go to the lymph node, they train the T cells, the T cells go back, they talk to the cancer cells, and then, in the best case, they kill the cancer cells. And if the case is much more better, they have a sustainable answer because they have memory produced by the immune system. Why does that not happen? Yeah? Because to make this happen, we need a plethora of factors we have to keep in mind. So the robust immune response is that what we want to have after perhaps the combination of radiotherapy and immunotherapy or after immunotherapy itself. And for this plethora of immune modulators, we need the understanding of patients' immune phenotype because it's crucial for optimization of the trials and the therapy-based immune modulation. But why is this so? And I want to show you one very, very old slide. It's now 21 years old. And that's the same what I show my students when I talk to them, and I want to know that they know how immune therapy is working. Normally, our body can be rid of cancer cells, of modulated cells. But the cancer cells try to survive in the body. And then the immune editing concept takes its place. And at the end, we have an escape. And the escape is not that we train one immune cells and we train two immune cells, that they suppress and that they leave the cancer in the body. we have the tumor trains the whole immune system to be suppressive against the tumor. And that's we have to become rid of it to get a strong and robust immune answer. So you talked about the industry that earn billions, and the immunotherapy industry, they earn also more than 1 billion with some molecules which they sold for immunotherapy and starting from the 2010s. And these inventions are coming not from the oncologists, these are inventions from the rheumatologists. They use these molecules 10 years longer than the oncologists. We have one molecule, it's mostly the co-stimulating ligand we can block with CTLR4 antibody. We have in the lymph node, and at the tumor side, we have the PD-L1 ligand axis we can block. And it works for many patients, and it works for more patients if we add radiotherapy to it. We have heard from the Pacific trial, and we know the keynote trials where we showed that 20% of the patients live longer, and 20% of the patients have a better effect when we add radiotherapy to the immunotherapy. But for understanding these things, we have to have knowledge about the immune system of the patient. And I heard about the lymphopenia, and they said, okay, it's not only lymphopenia, it's a matter of the changing of the immune composites, and therefore we are doing this immunophenotyping. And I was searching for papers for Hadrontherapy or combining therapy, where they have also done several things. And I've come to one, it's on bioreservics, it's not yet fully published. And they see more or less, it's a really small amount of patients and a different entities of tumors, and they see more or the same what we see also with photons, especially the T cells. They are decreasing after the therapy, but they are increasing at the end of the therapy. And that's very important, yeah, because they are not lymphopene at all. The T cells are coming up again. This was also shown in the 90s, and no one believes that the radiotherapy, when you go to a hematologist, he says, with radiotherapy, you kill all the immune cells. Yes, he wants to kill them because he wants to have a bone marrow transplantation afterwards. But you killed cells, but they recover. And then it's very important that these recovering cells know nothing about immune suppression from the tumors. That's very, very important. And therefore, we have to understand not only the T cells, we have also to understand the rest of the immune cells. They are only sticking in their paper, that nice paper that has several flares, which I want to discuss, but it's a very nice idea to look at it. And that's bring me to my next challenge. The next challenge is the characteristics of the tumor. And I just pick up two of them. The one is the resisting cell death, which is driven by the tumor, and the senescent cells. I will not come to the polymorphic microbes, also known as the microbiome, which you have to deal with when you're treating head and neck cancer. Yeah? I just want to stick to them. And I think you have heard a lot about low LAT and high LAT effects. I'm not a specialist in DNA biology, so I will not go in detail with this. But I have seen at a very, very interesting review that you change the cell death type. And it's a very, very important matter for the immune system, because every dying or dead cell will be taken up by immune cells. And then they have to decide. And a colleague of mine has this a very simple depiction of this, have shown what happens. Apoptotic cells normally lead to tolerance induction, while necrotic cells can lead to several other things, normally to a stimulation. And senescent cells, we don't know what happens to them. They live again, or they die at the end, they die like a necrotic or apoptotic. So it is very, very important to know what happens when I hit a cell, what happens to the cells. They are just stressed, they go to senescence and go back again. They are dying by apoptosis, necrosis, or several other forms of cell death we know today. I think normally they are defined 8 to 10. They are really defined as form of cell death, and they have all, it's very nice depicted here, huge amount of surface molecules and also soluble factors like chemokines, danger signals and something on which can drive the immune answers or can suppress the immune answer. Yeah, that's very important, and that is also a point which is not so big thing in particle therapy. We know about the cell deaths very much, and we know about also the reviews from Marco Durante and colleagues, but the thing what happens to the dead cells is very, very important, because the dead cells are the driver for the tumor microenvironment, and they have to shape the tumor microenvironment that the immune cells can come in and kill the tumor cells. So we think there is perhaps a magic dose. This was a paper from Menti group. They show the Zika sting pathway. They show 8 grays 3 times is the more or less immunologic dose. We don't see this in all instances like they see it, yeah? But they also say a high dose activates tracks, and we have no cytosolic DNA. That's at the moment the theory, they explain it, and then they have immune suppression. So that's another point. We have the dying cells, and we have the dying cells, they have to die correctly. So high dose means not only that all cells die and all cells can start an immune answer. And for this, we have perhaps to add, for some radio-resistant tumors, enhancing molecules. I found here a very nice paper some months ago, and they added a nanoparticle to enhance the effect of radiation therapy. We do this also with normal photons, and we see very, very interesting effect. And we see we perhaps kill more cells, but the cells we killed with this radio sensitizer, they are not so immunogenic than the cells we killed without the radio enhancer. So that's another point we have to look for. So, but at the end, I think particle therapy is a very, very broad field, and I think it's a big future field. And I have many of therapy options today, and I think we can go further in analyzing several things. I looked to some, I think, very potent reviews the last month, and they also claim that cytosolic DNA plays a significant role in the immune response. I agree with this. And this is also drives the tumor microenvironment. And very, very important, we need a bioindicator or biomarker. And that's not only DNA sensing biomarkers. These are also immunologic biomarkers. Look to the immune system if you want to understand how a patient is reacting to this or another therapy. And here is also underprinting identify more widely available to patients. That is, for me, I'm thinking a big problem and a big issue, that you have several centers, and that's also a big issue for research. So we can do with, at Erlangen, we're doing our preclinical models with our typical LIDACs. We develop many of things where we can irradiate mice or rats with our typical LIDACs, and we provide this also to partners far away, so many of the people can do their research. I think that's a little bit a drawback from particle therapy, that they have to go to several centers. I was also here at Garnil several years ago for glioblastoma, and that perhaps is a little bit a drawback for research in particle therapy. But I think the challenges of combination therapy for hadron immunotherapy is that we have to know how hadrontherapy is shaping the tumor micro as well as the macro environment, meaning also that you perhaps banking blood or doing immune phenotyping or looking for the blocks from the pathology if you do the trials with the patients, yeah? And I think also DNA damage is crucial for cell fate and cell death, and especially for the clearance of the cells being hit. Yeah, that's very important. Also, not only look on the hit cells, but also look on the surrounding. What happens to the cell if they are going to die? One big thing which is also under discussion at the moment in normal radiotherapy is volume reduction in radiotherapy always the ultimate goal? the role of the lymph node is actually under current research. Some people say it's you have to spare them. Others say you cannot spare them because perhaps there are some tumor cells in and they get metastasis. And I think would hadrontherapy be valuable for combination of bridging therapy with cellular immunotherapy? That's what our move at the moment, that we go to the people who are doing cellular immunotherapy as a bridging technology. There was half a year ago a paper from the US showing that when they do the bridging therapy with radiotherapy, then the CAR T cells react much more efficient to the tumor than when they're not doing this. Another hint that the immune system can be modulated by radiotherapy as well as particle therapy. And I think there are more specific radiosensitizing enhancer working significantly better with hadrontherapy. I think not all the radiosensitizing enhancer will work with hadrontherapy in the same line as they work with photon therapy, since the damage of the DNA is a clear difference. Yeah? But I think I stick to the mind of Marco Durante. Heavy ion therapy can modulate the immune system as well as photon therapy can do it. So then I thank you very much for your patience, and if there are questions, I'm open to it.
We can have one burning question.
In case I have questions.
Angelica.
You wrote that DNA damage from carbon ions and highlight inhibition is crucial for the immune system. What do you think about the damage to other organelles or part of the cell, for example, cell membrane? I mean, we always focus on DNA damage. That's fine.
Yeah, we always focus on DNA damage, and that's the same what the photon people do since years. Yeah, and we came to it and we say we are not interested in DNA damage, and said you're crazy, you're a radiobiologist, you have to be interested in DNA damage. I think DNA damage is one part of the damage to the cells. The special point is how perhaps is this repaired, yeah? Or how can I stress the cells in advance? So we know especially stressed cells with a huge DNA damage are the most immunogenic cells. Especially they have endoplasmatic reticulum stress.
Yeah. Or membrane or
stress or mitochondria.
Yeah.
There are several other organs than just the nucleus.
Okay. So in principle, if you go like with the high dose and so on, you can have like some cell death. For example, you have HMGB1, ATP release, and so on. If you go with the lower dose, perhaps you can increase the reticulin. And then let's say having a double dose into the tumor, perhaps could be also a good, let's say, rationale. For example, what do you think about the PATI, so this partial tumor irradiation from Slavisa, for example? This should increase like the immune response, or?
I think it could enhance the immune response since it enhances the cellular stress. And the stressed tumor cells is always a good immunologic target. So if I kill a stressed tumor cells, I have the possibility to release all the danger signals to the tumor and shaping the tumor microenvironment to a more immunogenic or inflamed tissue. And then the cells can come in and can kill the tumor cells.
If I'm right, you mean that a bad radiation oncologist could be a good radiobiologist?
I just ask if he thinks that the partial tumor irradiation is something that...
So, unfortunately, I see that there are several other questions. We should keep the questions for the discussion. We have to go further for the next one. Thank you very much, Benjamin.
Thank you.
So the next speaker, Professor Iuliana Toma-Dasu, who is the professor of radiation physics at the Department of Physics, actually, at Stockholm University and Karolinska Institute. And well, you have been working a lot with the hypoxia and modeling the hypoxia, and you are welcome. Maybe you got answers to some of the questions that you had in the previous discussion. Please.
Or more questions to add to the previous discussions. So thank you very much for the invitation, and thank you for inviting a medical physicist to talk on a radiobiology session about such a fantastic topic as hypoxia. So of course, I have to excuse myself from the very beginning for not being an expert, but only giving you my biased view as a medical physicist. So I don't have to state more than hypoxia is bad news in this context. We know, of course, that hypoxia or absence, limited oxygen supply leads to poor local control, but also poor long-term prognosis, because hypoxia leads to resistance to radiotherapy, as we know, and also resistance to chemotherapy because of the malfunctioning vasculature, and not only, and because of being prone, the hypoxic tumors to form local metastasis and then to spread local regionally. It's bad news for poor long-term prognosis. What we know about hypoxia from preclinical in vitro studies, it's summarized in plots like this. A dose as much as 3 times higher, it's needed in hypoxic conditions, in anoxic conditions, to eradicate the same proportion of cells as in oxic conditions. But of course, this hypoxia is not a black and white picture. We have many levels of hypoxia in between, and we know that the relative radiosensitivity is depending on the oxygen partial pressure in the tissue in a sigmoid manner, as illustrated here, that depends on the partial pressure of oxygen, as I said. So knowing this now for more than 100 years, what do we do in practice? Well, as we just discussed, much too less. Not only with particles, but also with photons. And I always, when I give a talk on hypoxia, touching on the role of oxygen and the way of radiotherapy should address hypoxia, I like to quote this paper by Jens Overgaard, because it has a very catchy title that says a lot. Hypoxic radiosensitization, adored because we love to talk about it, and ignored because we do much too less in comparison to what we should do. Although we have many ways of tackling hypoxia to some extent. And there is a list here that was put up by Jens Overgaard about ways of sensitizing the hypoxic cells or hypoxic tumors. To that I added hypofractionation, dose escalation based on hypoxia imaging. The list is of course not exhaustive, but I would like just to stress that Jens Overgaard wrote there explicitly, elimination of OER by high LAT. These are very strong words. I don't particularly agree to the elimination of OER as such by high LAT unless we really define it. Few years later, we have the review by Walter and Marco that was also talking about the advantages of hadrontherapy in this context. But there, the way of phrasing this potential advantages was more nuanced, saying that, well, hadrontherapy could be effective against hypoxic tumors. Effective is not the same thing as eliminating, eradicating completely the OER issue. Do we have clinical evidence? Well, we discussed about this the whole morning, about the different clinical evidence that it's available. But as it was pointed out, not so much for treating hypoxic tumors. And perhaps one of the most quoted studies about the clinical evidence that carbon ions are effective against hypoxic tumors. It's this study that I put up here, in which the oxygen partial pressure was actually measured by an Eppendorf electrode in tumors, and the tumors were stratified according to the oxygen partial pressure. And there is a comparison here between the effectiveness of carbon ions in hypoxic and oxic tumors. Measure this oxygenation in the beginning of the treatment or after 5 fractions. And the photon arm in which the same was done. And what was observed there and taken as a proof of the effectiveness of carbon ions on hypoxic tumors, it's this overlap between the curves. So we can see that for oxic and for the hypoxic tumors, pretty much the curves for carbon ions overlap. Whereas we can see that for photon therapy, of course, there is much less effect on the hypoxic tumors, but for the oxic ones, they are doing quite well. It has to be noted, however, that here we are comparing a little bit orange and apples, because these two treatments were not involving the same number of fractions, not the same BD. Then there is indirect effect. We know, as we discussed, that hadrontherapy is effective against pancreatic cancer, known for its radioresistance. It's effective in glioblastoma, again known as radioresistance. But there are not so many studies in which the hypoxia was actually measured. And perhaps it's time to build up more clinical evidence. Now, this is the paradigm. Hadrontherapy, it's able to control hypoxic tumors, and it comes from these overlapping cell survival curves for oxic and hypoxic irradiation conditions. Then, if we look a little bit careful at these curves, yeah, it's correct that they overlap, but the LAT for which they have been determined, derived, it's really very high. So maybe we should question this paradigm. Because in reality, in a spread out Bragg peak, we are not reaching these very high levels of LAT. And yeah, this is just an example. In a typical SOBP, we will have something between 30 and 80 kiloelectron volts per micron. And that would lead to an oxygen enhancement ratio between 2 and 3. So in theory, in the lab, high LAT particles would, of course, eradicate hypoxic cells. In reality, they will also eradicate hypoxic cells. It's just that we don't deliver this very, very high LAT. And this was also very nicely summarized in a paper, in a study by Walter and his colleagues, not only for carbon ions, so for different species of high LAT particles, for a very broad spectrum of oxygenations. And what was shown there is that indeed we could get an OER that is close to 1 if we have not hypoxic conditions, but rather well-oxygenated cells, or the LAT is very high, or in combination, of course. So let's ask the question in this ideal way. We have a patient, and we can determine the presence of hypoxia. What treatment do we choose? Do we choose a photon treatment with a conventional fractionation, or maybe protons with conventional fractionation, relying on the reoxygenation, or we go for carbon ions, hadrontherapy in general, with hypofractionation, aiming to deliver high LAT, or perhaps a very highly effective dose in one single fraction? Well, there are more questions to be associated to this question. First of all, on the imaging part, on determining actually the presence and the extent of hypoxia. What biomarker do we use? What is the timing? What is the quality of the image? How we go from, let's say, it's a PET image, SUV to PO2, how we discriminate between different levels of hypoxia, because as I said, it's not black and white. How we capture the dynamics of hypoxia. Some of them are working in our favor, this reoxygenation. What is the optimal fractionation schedule? What is the optimal treatment plan, with everything that means, including the choice of the particle type, fractionation, and so forth. Now, of course, this is a radiobiology talk, and I will talk about something connected to radiobiology in silico trials. So using the knowledge from radiobiology to create virtual tumors that could be irradiated in virtual in silico trials, hoping to get some answers, or at least, well, moving in the right direction with respect to answers to these questions. So I will show you some examples of the results of some in silico trials in which virtual tumors were irradiated with photons, protons, carbon ions. Today I will only show examples with photons and carbon ions. Different fractionation schedules to determine, to calculate the relative clinical effectiveness and not the relative biological effectiveness, because RBE is defined for the same irradiation conditions except for the quality of radiation, meaning the same number of fractions. But we are not interested in comparing a type of treatment like Hadrontherapy that should be delivered probably in just few fractions with photon radiotherapy, that it's more likely. I mean, this is what we would like to compare. I mean, hypofractionated Hadrontherapy schedules with conventionally fractionated photons. And in that case, the RB is not valid, so we will have to look for a relative clinical effectiveness. And we should also look for a clinical oxygen enhancement ratio, not this that compares the effectiveness of Hadrontherapy on hypoxic tumors, so not just hypoxic populations in vitro, so different degrees of oxygenation. So this is what we have worked on. We simulate virtual tumors with respect to angiogenesis, and we could get something like this in terms of resolution in oxygen partial pressure, so at microscopic micrometer level. So we could simulate the diffusion-limited hypoxia, so the chronic hypoxia. We could also simulate anemic and diffusion hypoxia, also by pruning some of this vascular tree. We could simulate acute hypoxia, so perfusion-limited hypoxia, and more important, we could simulate the dynamics of hypoxia, reoxygenation, local reoxygenation due to these changes in the oxygenation at cellular level, as well as global reoxygenation, because we know that tumor hypoxia, it's not a static feature. It's not what we see in the beginning of the treatment that we have all through the treatment, because if that would be the case, then probably we would not be able to really eradicate hypoxic tumors. And we have worked quite a bit on this dynamics of hypoxia with PET, monitoring changes over the course of the treatment. But this is also, I mean, this would be at macroscopic level, but at microscopic level, there is evidence that this hypoxia is dynamic. All right, so if this is the case, we can simulate in silico trials, or we can conduct in silico trials. We can implement the model for the tumor into a treatment planning system. Then we simulate the treatment, and we calculate the probability of controlling the tumor. And this is an example for a well-oxygenated tumor that is treated with photons delivered in 30 fractions. This is the TCP. If we change to 3 fractions, we move, of course, to lower doses. We go for a hypofractionated treatment. We can do the same for carbon ions, 30 fractions, 3 fractions, and in this way we can determine a relative clinical effectiveness as the ratio of the doses leading to 50% control at the same level of control for a treatment with photons delivered in 30 fractions relative to a treatment delivered with carbon ions in a hypofractionated manner in 3 fractions. And an RC in this case will be 4.5. We can do the same for a hypoxic tumor, and in that case, the RCE, the relative clinical effectiveness of carbon ions delivered in 3 fractions, will be higher, 5.4. So if we compare this, we can conclude that larger relative clinical effectiveness would be expected for hypoxic tumors. I'm not saying large or control of the tumor, only the effectiveness. So here we talk about the ratios for 50% TCP. Now, this RCE, as I said, will increase for the treatment delivered with carbon ions in few fractions in comparison to typically fractionated photon treatment, and it would be the highest for tumors that are persistently hypoxic, so those in which these local changes of oxygenation are not that frequent. We can define a clinical OER if we are now pairing hypoxic tumor control probability curves with oxic tumor control probability curves for the same type of particles and the same number of fractions. And we can observe that a clinical OER will also be far from 1, even for carbon ions, depending on the number of fractions, if we talk about these persistent hypoxic tumors, but it will go down to closer to 1 if these local oxygen changes will take place. And this is just a summary of the results, pointing out that the relative clinical effectiveness is higher for persistent hypoxic tumors, and the OER is far from 1 in clinical terms, even for particle therapy. So in summary, hypoxia, it's still a problem. It's still a problem for hadrontherapy, but there is a lot to learn from what it's done in terms of dose painting, escalated, simultaneously integrated boost in the world of photon therapy. There is a lot of effort put there into integrating imaging in the treatment plan with respect to determining the features that are related to poor outcome in the beginning of the treatment, to optimize the treatment, but more than that, to adapt the treatment, coming to what Piero was saying. There is a lot of there is a lot of advantage expected from integrating functional imaging into treatment adaptation. And that has been demonstrated for photon radiotherapy. It's just a matter of looking closely into that and adopting it for hadrontherapy, because it appears that it's needed. And now we have these tools developed for photon therapy. We have the tools for optimizing the treatment plans in a multi-ion beam manner. For ion therapy, it's just a matter of letting the two worlds to get together. So thank you for your attention, and thank you to my collaborators.
Very short question if you have, while you are changing the file. Thank you very much, Alina. So, I would like to welcome Claire Rodriguez-Lafrasse from University of Lyon, who is very focused on effect of different LET and molecular mechanism of different LET radiations, and to give a summary of your work. Thank you.
Okay, thank you. Good afternoon, everyone. Our research project is called BioHadron. I started working in radiobiology in Lyon in the early 2000, and Professor Jean-Pierre Gérard was the dean of my faculty, was radiotherapy. And he told me about revolutionary radiotherapy technique developed by Japanese. And he said, we need it in France, and we need a radiobiology team in Lyon. You should go for it. And I took out this challenge, and I joined the Étoile project from the start. So at the beginning, at this period, Japanese showed very nice clinical results, but there was not enough randomized clinical trials to determine the benefit of carbon ions compared with conventional radiotherapy, and there was a clear need of scientific proofs of the superiority of carbon ions. So the question we were asking us was, are there specific molecular mechanisms involved in the response to carbon ion, and do they more effectively kill radio-resistant tumor cells? So over 20 years, we radiated tumor cells and animals at different facilities in Gani, GSI, and IRS, CNAO more recently, at different LET, and we accumulated extensive data which enable us to formulate the stealth bomber paradigm. this paradigm became evident to me when I compared our results to the simulation performed by our colleagues Michael Beuve and Caterina Monini. They simulated the hydroxyl radical distribution at 10⁻12 seconds in the nucleus volume in response to 2 gray photons or carbon ions at a physical or biological equivalent dose. And it's very clear from this simulation that hydroxyl radicals are distributed uniformly in response to photons, while they are distributed along the tracks in response to carbon ion. And you easily understand that the consequences at the cellular or molecular levels are very different. And this led to the stealth bomber paradigm. So I apologize for the military analogy in the current context, but it's perfectly explain and illustrate this concept. So it is composed of a bomber effect when the subcellular targets are on the targets of reactive oxygen species or carbon ion tracks, and the Ross-induced complex damage at the DNA at the molecular level. At the opposite, the stealth effect comes from the fact that very low Ross are produced out of the carbon ion tracks, and this leads to the absence of triggering of cellular alarms and defensive mechanisms outside the areas hit by Ross. So I will show you our experience that support both effects. So the bomber effect is very intuitive and has been described largely in response to DNA damage. So to summarize, we found and we confirmed that carbon ion induced complex DNA lesions that are difficult to repair. We also showed that the chromosome loss is a specific signature of carbon ion irradiation, which is not transmissible to the progeny and thus decrease genomic instability. And finally, we showed that the killing is independent of the telomere length. So as said, Angelica, DNA is important, but other compartment too. So proteins are very important in the cells, and we showed here, perhaps I can put this. We showed that there is an increase of oxidized protein in response to carbon ions. The color code will be blue for photon irradiation and red for carbon ion at biological equivalent dose. And these increased oxidized protein are not taken in charge by the classical plaque pathway that are the proteasome and the UPR pathway. At the cellular level, the consequence is more cell death, and the cell death is triggered earlier and is more important in response to carbon ion at biological equivalent dose. And it's important to highlight that they better kill cancer stem cell, and this cell death, there is no specific mechanisms involved. Another consequence of the bomber effect is that the cell killing is independent of the oxygen concentration in our experiments, which is favorable for the treatment of hypoxic tumor, and is also independent of the radiation dose rate, which is important for the planification of treatment. So now let's focus on the stealth effect, which is unique to carbon ion and is much more exciting, as I told you before. It comes from the fact that a large proportion of the cell volume at the nanometric scale is not hit by carbon ion, so the threshold of frost necessary to trigger survival and defensive mechanisms are not reached. And this image of the stealth bomber illustrates the fact that carbon ion cross cell radars and lines of defense without being spotted, and I will show you some important results. So the first event in the chronology of molecular events is DNA damage detection, and it is delayed, as you can see here on this graph of the nucleotide shuttling of ATM. And Malouf et al. Describes that in response to oxidative stress, ATM monomers are formed and translocate to the nucleus to detect double-strand break, and it is delayed here in response to carbon ion irradiation. So the consequence is also a decrease of signaling and DNA repair. This is illustrated here by three biomarkers, gamma H2AX for signaling, phospho-DNA PK for NHEG repair, and RAD51 for homologous recombination. Another important point is also the absence of stabilization of IF1 alpha. You know that IF1 alpha is a major transcription factor involved in the response to hypoxia, but it's also stabilized by photon irradiation, as you can see here. And this is not the case in response to carbon ions, and this can partly explain the absence of oxygen effect in response to carbon ion, but also the absence of several adverse effects depending of IF1 alpha, such as angiogenesis, cell proliferation, and invasion migration. So this is further demonstrated here for invasion and migration. Here you can see that photon irradiation increase invasion and migration in cancer stem cells. This is not the case in response to carbon ions in red. And we showed that the mechanism involved the absence of activation of the three main signaling pathway involved in the epithelial to mesenchymal transition, but also in the P5038 MAP kinase pathway. From a mechanistic point of view, we also showed a significant decrease of MMP2 concentration, which is involved in the degradation of the extracellular matrix during invasion. And we also recently showed by atomic force microscopy that carbon ion irradiation increased the rigidity of cancer stem cells, and this significant decrease of cell plasticity could explain their lower capacity to invade and migrate in the surrounding tissue. We also studied the stress granule formation. These are non-membrane cytoplasmic aggregates which are formed in response to oxidative stress, and which can protect mRNA. And here you can see the double leveling of a protein on mRNA, which are specific of stress granule. You can see here stress granule formation around the cell in response to photon irradiation. And there is no stress formation in response to carbon ion in all the conditions tested due to their specific Ross distribution. So to summarize, the Bomber effect explains the high RBE and their ability to overcome radio resistance, while the stealth effect prevents unwanted adverse effects typically associated with photon irradiation. So what about the immune response? So we talked this afternoon about the immune response. To answer this question, we developed an immunocompetent mouse model, and they were grafted with head and neck tumor. I didn't specify before that we largely work on head and neck tumors. And we compared 3 protocols of irradiation with photon, proton, and carbon ions. It was a fractionated regimen with a biological dose of carbon ions, and it was combined or not with immunotherapy. So to summarize the result, we showed a higher efficacy of carbon ions at the biological equivalent dose compared with photon and proton. We also found a higher vaccinal effect with carbon ions. Concerning the combination with immunotherapy, we found comparable results between the 3 modalities of radiotherapy, but we found an increase of immunogenic deaths with proton and carbon ions. So I hope to have convinced you that because of the stealth bomber effect, carbon ions have biological superior effects to the best conventional radiotherapy technology, particularly to cure radioresistant cancer. And we are testing this effect with other young. We hope to confirm this effect, and it's also more important that the cyclotron in Caen will enable treating patients with other ions. Thank you for your attention.
Thank you very much, Claire, for this very nice summary of the work you have done. So any burning question to Claire? Yes, please.
From Hadrontherapy. What about inflammation on LC tissue? Do you have any data on that?
LC tissue?
Yeah,
no,
tissue.
I didn't have an experiment with LC tissue. We have collaboration with IRSN, but I'm not sure they observed a difference between both. I can't answer you.
Yes, so we continue with 6 more presentations. So as you see, the schedule is very tight, and I would like to ask the speakers, please keep the time, and then we try to, if no time available, so then we have this opportunity to have your question in the discussion later on. So I would like to ask Walter Tiganelli, my co-chair, to give the first talk. And Walter is from GSI. Everybody knows him, I guess, after so many...
I don't think so. Well, thank you. Hello, good afternoon. Thank you very much to the inviter, for inviting me here today. It's a pleasure for me. So I will speak about ongoing research activity at GSI Radiobiology Lab. And then I've been told to stress a little bit the point of the flash carbon that we are doing at GSI. So I will also present a few slides about that work. So essentially at GSI we perform experiments that are about physics and natural science disciplines. In GSI between 1998 and 2007 there was the first carbon ion treatment of a patient. That was September 98. They were the first patients treated for a period of 3 weeks. Nowadays, we are not anymore a clinical facility. Actually, this is one of the questions that we have, I mean, most of the time. So we are not anymore a clinical facility. We have experimental rooms. Of course, we can accelerate from proton to uranium. We can go up to 1 GeV, the energy range. Beam delivery, we have static and pencil beam. Active and passive energy modulation. We have no gantry. And we are allowed to do preclinical studies. So we are doing actually nowadays more and more mice, rats, and we also had pigs in the past. How to apply to GSI beam time? Essentially, GSI is a user facility. So we have to allocate beam time to the external user. That's why we have 2 different packs. One is the biopack that is for experiment in biophysics, radiobiology, radiotherapy, and the biopack meets every 2 years. And then we have another pack that is the HIPER. So GSI is the ground-based facility of the European Space Agency. And then if someone wants to do experiment related to space radiation, can come at GSI and perform the HIPER experiment. At the moment, we do not have so much beam time, and the point is that we are constructing the FAIR. So you can see GSI is this one here. You can see also here. And now this is a drone view of the FAIR. So it's like a new synchrotron that will give us the possibility to go to very high intensity, very high energy. And in the next years, in the next couple of years, we should start to use the facility here for the irradiation. And then, yeah, GSI will become essentially GSI FAIR. For those people coming from outside, when they come at GSI and they want to use our facility, essentially there is like a lab that is available for them. So we have the animal house for mice, animal house for rats. We have a biochemistry, cell lab, microcity for animals, and then immediately close to this lab there are the cave M, cave A. So the 2 rooms where actually we can irradiate cells, organoids, animals. And in the cave M we have also a SARP machine, so we can also do study with high precision X-rays. For mice we have like 300, the maximum that we can guess is 385 mice at the moment. Rats, we are improving our animal house and we can go to 60, 75 rats because, I mean, this depends a lot on the dimension, if you can put 4 or 5 rats together in the same cage or not. In the biophysics department, we are essentially 9 different groups. So 5 are about biology, radiobiology, and 4 are about physics, medical physics, radiation physics, space radiation physics, and so on. I asked all the group leaders from the other group to just give me 1 slide because I want to show what is going on at GSI. And then in the end, I will present some work that we are doing in the clinical radiobiology group. So the first is the molecular radiobiology and imaging group. So the group leader is Burkhard Jakob. Naturally, when we go to the conferences, we always present this nice video and nice pictures where we have the nucleus of a cell traversed by ion, and then you see this DNA repair molecule that occur to repair the damage. Essentially, this kind of work is done in the group of Burkhard Jakob. So they do spatial and temporal aspect of radiation response, charged particle irradiation, dose and lesion distribution, induction and repair of complex DNA lesion, and so on. So LAT, systematic repair, protein kinetics, and binding, and so on. Then there is the group of Insa Schroeder that does stem cell differentiation and cytogenetic group. Just to say, the group of Burkhardt is the group that actually is working with cells. Then we have organoids, that is the group of Insa Schroeder, and then you will see that the other three groups, essentially, they are working with in vivo experiment with animals. So the group of Insa instead is using human organoids, but region specific, and actually she has two different fields, let's say, of research, but this is something that is very common also in the other group. So we do either space radiation and cancer research. So we do both, and we collaborate with the other group in both the different topics. So radiation therapy, so she does tumor response to radiation therapy, optimal treatment modalities, side effects, and space radiation, she already had like parabolic flight campaign. Now she are going to send some of these organoids in the International Space Station, but she does impact in long-term mission and countermeasures. Then there is the group of Charlotte Van de Voort that is a pretty new group, so a space radiation biology group. Since the group started a couple of years ago, 3 years ago, I think, they at the moment have like a gut microbiome as a marker for space radiation risk. Then they do a big collaboration with us. We have a project on synthetic torpor or synthetic hibernation, and they study the gut changes in synthetic torpor. And then now still with us, I mean, we are doing the neon flesh. So the neon flesh now is the possibility now to use a very heavy ion flesh. So to go back, let's say, to the idea of Tobias in Berkeley, to use very heavy ion to treat the very radioresistant tumor. So at that time, they did some trial, and they found out that actually with neon, you are able to kill even the very radioresistant tumor, like the glioblastoma. But then, of course, you have a lot of toxicity in the surrounding tissue. And then the idea now is, if there is a sparing effect with the high LAT, ultra-high dose radiation, like with carbon, perhaps we can check if there is also something with neon, and then perhaps neon can be used for this kind of treatment. And I will not speak about the neon flash. This is something that we are going to do now, essentially in the next biopack application, but I will be concentrated more on the recent results that we have on carbon flash. Then there is the group of Claudia Fournier that is essentially working on immune system and tissue radiobiology. Then, of course, they work the combination of radiobiology with checkpoint inhibitor, but they also had like an important study in collaboration with BioNTech, studying the effect of RNA vaccine that we all know, I mean, during COVID, RNA vaccine and cancer in combination with carbon and X-rays. So there is here, I think, a couple of, where you see Nadia Salomon is from the group of BioNTech, so you can see some of their publication. And then there is the clinical radiobiology group. So in the clinical radiobiology group, we are working with flesh, heavy flesh, let's say. So we are using carbon, but again, we would like to move also with neon. Then we have synthetic hibernation for space exploration and medical application. We actually have a lot of interesting results showing that synthetic hibernation, so a physiological phenotype that resembles the natural hibernation, is able to increase the radiation resistance. And now we are testing this kind of phenotype also with medical application, like for cancer research. Then we have effector radiation on circulating tumor cells. So circulating tumor cells are those cells that from the primary tumor, they detach, enter into the bloodstream, they reach a distal site, and they form a metastasis. We want to see if different kind of radiation, different LAT are able to produce a different phenotype and number of circulating tumor cells. And then we work constantly in tumor hypoxia and developing new phantom to test the hypoxia in vitro in our lab. About flesh, so as I said, now I'll stress a little bit more this point. About flesh, we all know what flesh is, but just let's say a small summary is when you have like an ultra high dose rate, so when you have like more than 40 gray second compared, let's say, to the 1 gray minute or 2 gray minute that is used in conventional radiation, you have like less than 200 milliseconds is the dose that is delivered. High dose in a single fraction, even though now I spoke with colleagues from Japan, I don't remember now which university, but essentially they say that, and also colleague from Ganil, they say that essentially even with a low dose, they are able to see a flash effect using carbon. So essentially what happened in this case? You are able to spare the healthy tissue and control the tumor growth as much as with the conventional dose rate. So essentially you are able to spread a bit the therapeutic window, moving the normal tissue complication probability, let's say, on the right. So we had the idea, after a lot of experiment with proton, electrons, and so on, to test also flash with carbon. And actually the first experiment we did in collaboration with it, actually with Ivana, that is here in the audience. And here actually we use this hypoxia chamber that we have at GSI. So essentially inside here this is a GAF chromic. So you see the 7.5 gray that are released on the GAF chromic here. And then here we had our cells that are at the desired oxygen concentration during irradiation. So you can gas this chamber, and the chamber keep the oxygen concentration during irradiation. So we tested at that time different oxygen condition, because we believe that at 21% or at the 0% of oxygen, we would see no flash effect, but in the intermediate oxygen concentration, like 0.5, 4%, we would have seen something. And actually these were the results that we published in the International Journal Radiation Oncology. And then you can see here, this is simply the survival of CHO cells, Chinese hamster ovary cells. And then you see 21% flash, conventional, no differences. There is also another study with 0%, but for matter of time now I'm not showing this. Then you have 4% flash, you have higher survival compared to the conventional, and 0.5 you have even higher survival compared to the conventional one. Then we decide to move to the in vivo experiment, and actually we designed a 4-phase protocol for testing flash in vivo. The first phase was also in combination with DKFZ and HIT. I have to stop the music here because otherwise we will hear this sound forever. You know how to stop it? Otherwise, looks, I already said this like a submarine. This sound like, okay. Nothing. Okay, you have to, we are close to the ocean, it's nice. So, in this first protocol, essentially, we just want to irradiate the thorax of black 6 mice, so no tumor, nothing, because we want to see after 6 months the lung fibrosis. So essentially, here we are irradiating just normal animal, no tumor. Okay, 1 minute, I see. And with a plateau and gray, and then you can see here lung density, lung volume, fibrosis index, survival. Essentially, you can see that flash is always better than the conventional. Fibrosis index, you have more with conventional, lung density more with conventional, lung volume is more with flash compared to the conventional, and also the survival, you have higher survival of the flash animal. This is a manuscript in preparation that, again, now our colleague in DKFZ are writing. Then we did another experiment where essentially we irradiate both the posterior limb of C3H mice. In one of the limb, we injected the murine osteosarcoma LM8. So we irradiate both of them, and then we check the muscle of this limb just to check the sparing effect of the ultra high dose rate. And on the other tumor, on the other limb, we check for the tumor control. Then you can see here, I mean, this is the control. Of course, the tumor is growing during the time. And then here you have flash and conventional. They both control the tumor growth. When you check at this other limb, the one without the tumor, we see that flesh is preserving the muscles compared to the conventional one, where you have a disruption of the muscle itself. And then in this first paper, we found very interesting, this is entrance plateau, we found very interesting that you see this kind of tumor produce a lot of metastasis into the lung of the animal, and then in the control you have a lot of metastasis. Conventional you have a small decrease, but with flesh you strongly decrease the metastasis into the lung. Then we move to a second protocol in which again we had the tumor in one of the limb, again C3H, animal model, again murine osteosarcoma LM8, and now we irradiate both limb with the SOBP of carbon. Also in this case there is the same control, flesh and conventional. Also in this case there is a sparing effect. Now we check for the collagen deposition. You have a lot of collagen deposition after conventional compared to the ultra high dose rate. And in this case we had like a strong reduction of the metastasis, but this already with conventional. But this is a data that we already had, and Takashi, most probably Shimokawa, Dr. Shimokawa, will also present some results in Japan, where actually we had already this data. So conventional carbon is already able to reduce the number of metastases in this animal model. And now we have data not published. I mean, we are working on this since before there was the request about the abscopal tumor. I don't know how to repeat the not abscopal tumor in the other version. It was very nice, but I don't know how to repeat. But essentially here we had like C3H mice, murine osteosarcoma. We inject the tumor in both the posterior limb. We irradiate this tumor, and then we check for metastasis, tumor, and also for the abscopal tumor. We wanted to check if there is an effect. Now this is just few data, but essentially here is the irradiated tumor. So this is the control, only the CPI, the radiation, and radiation combined with CPI, so that is better than the radiation alone and better than CPI. But when we look at the abscopal, now it's a little bit a confusion. I mean, we do not understand yet. I mean, we are studying now. But it looks, I mean, even though here looks like not significant, it looks like the conventional plus CPI is even better than flesh. But this is something that now we are investigating. When we check at the immune infiltration, this is the last slide. When we check at the immune infiltration, let's say these are the good cells, these are the bad cells, this is the primary tumor, this is the abscopal tumor. Essentially, there are no differences. CPI, it looks like the CPI is the one that is very important in increasing the population, let's say, of the good cells into the tumor. The only difference that we found in the irradiated tumor and in the abscopal is the Th17, that actually they are good and bad cells. Also this, I mean, something that we need to clarify. But you see here in the primary tumor, and here in the abscopal tumor, you have an increase of this Th17 conventional plus and minus using the combination of anti-CTL4 and anti-PD1. And with this, I conclude. I thank you, and I thank the group and the GSI Biophysics Department. Thank you.
Thank you very much, Walter. While we are changing the file, is there any burning question to Walter? Otherwise, during the gala dinner.
Okay. Yes, I introduced the next one. And now I have to find the paper first. Okay, and now we have Dr. Iuliana Dokic, Heidelberg University Hospital, Division of Molecular and Translational Radiation Oncology, Heidelberg Ion Beam Therapy Center, that will speak about heat lab and helium results.
Yeah, thank you very much, Walter. Thank you for the introduction, and thanks to the organizers for giving me opportunity to speak about our data in biology and our biological research in Heidelberg at HIT. So you have heard this morning already from Semi a little bit about the HIT and our center, and it's just because I already made these slides, so sorry if I bother you, I will go fast through this, that it's a synchrotron-based facility, and we have active beam scanning delivery using proton, carbon, and helium, since recently ions for the clinical treatment. And we have also oxygen ions as a higher LAT ions for the experimental treatment. And here you can see how it looks like, the schematics. I just try to use this, yeah. So you have heard and seen about the clinical part, and we come to the research part in biology. It's like this not so nice looking part here in the corner, but still very efficient. So we are using still the same sources, the same beam, and so on. It just looks a little bit smaller and not so classy like these nice patient rooms. So basically it looks inside like a classical biology lab or physics lab. So what do we do there as biologists at HIT? So one of our main tasks is to try to overcome the radio resistance of different tumors, and we actually do not focus on a single entity. So we are trying to target glioblastoma, we are trying to target pancreas, some of the lung tumor models, and we are working in vitro and in vivo. And to achieve this, we are basically trying to use the physical characteristics of different available beams. And we know that each of the beam qualities has different characteristics in the terms of lateral scattering, fragmentation tail. And basically, we take it for our biological research. We test all of this in the terms of LAT, different dose, and since recently dose rate, again, flash. And since very recently, from this year, we are doing the mini-beam radiotherapy with the different particles. So just to start with, it's also what you have seen previously from Joanna, and you heard in the other talks, is that with the different LATs, we have increased from protons, helium, carbon, and oxygen ions, getting higher in LAT. And we saw that based on the LAT, the complexity of DNA damage is different. So as you can see here, for example, for the proton beams, it looks like a small foci. These are gamma H2X foci that resemble a lot for the X-rays foci. And then we have with helium, they are getting more clustered, even more with carbon, and very clustered chain of the DNA damage foci presented here in this nucleus. So what, in addition to this complex DNA damage and this higher killing, what we also heard, again, before I said, so I just don't want to repeat too much, but we've seen that the higher LAT, so carbon beams and even more oxygen beams, their toxicity on the tumor cells is less dependent on the oxygen status, and they can target more efficiently invasive niche. And interestingly, what we saw in vivo was that irradiation, for example, in this case with proton and even more with the carbon ion radiotherapy, can lead to the formation of immunopermissive niche. So for example, if we focus on glioblastoma, that is one of the most resistant brain tumors, and it's highly heterogeneous tumor, highly hypoxic tumor, and it has a quite characteristic tumor-immune microenvironment, it's very difficult to target it and select the treatment that is efficient against such tumors. So what we are trying to do here, okay, let's see if we increase in LAT can do something more than just simple higher DNA damage, higher killing, but is there some benefit that we could get from the immune response? And this is what we've seen here, that actually we have a higher decrease of the cancer cells, decrease in hypoxic cells, that we used the pimonidazole stain to measure that. We have a decrease in glioma stem cells. For this, we use different stem cell markers. We have decrease in endothelial cells, much higher than after the photon irradiation, increase in the cytotoxic T cells, GR factor, and we have the microglia M2, so let's say immunosuppressive profile decreased. So after this, what we've seen is that basically we were able to control the tumor in 3 different glioblastoma, orthotopic glioblastoma models in vivo. So using 2 different syngeneic models, here we have SMA 560, this is a spontaneous murine astrocytoma in VMDK mice, and here we have GL261 in classical, mostly used black 6 mice. And what we could see here that indeed this was the physical dose of 15 gray, that with the carbon irradiation, the survival of the mice was at the highest. And this data we could repeat also in the human xenograph model. So this is NCH 644 cells. NCH stands for Neurochirurgie Heidelberg. So these are the cells that are derived directly from the patient that was been taken for the biopsy. The cell lines are then established in the culture and then injected in the mice. And we could see that even with this human model, we were able to to extend the life of such mice. What is interesting, as normally with the xenograph model, we are using the nude mice, so there is no T-cell response, and the effect was still present. And this is something that we have to investigate more. What else, besides the immune invasiveness and so on, could lead to such a response? And one of the points was here. There's something that I'm not sure how many of you who are not into glioblastoma topic are familiar with, is tumor microtube network that has been found by our colleagues to be responsible for the glioblastoma radioresistance. So this was from the department from Professor Wick and Winkler a few years ago. They seen that if they irradiate the tumor, here it was 3 × 7 Grays dose, that with the X-rays, there is a DNA damage, but then the surviving cells reconnect with certain microtubes protrusions, and they are very thin and long protrusions. They are something like less than 15 microns in the diameter, and they can be long up to 500 microns. So these are very, very long protrusions that we were observing also in the culture in vivo and in the patients later on. And when this kind of network is formed, these kind of tumors then is very difficult to kill. So then further research on these microtubes has shown that these glioblastoma tumors don't make only the tumor-to-tumor connections, but they connect also tumor to astrocytes and tumor to neurons. And basically, what happens through these networks, now there is a lot of ongoing research there, is that they are basically being fed by the tumor microenvironment, by the healthy cells. For example, for microglia has been known since really long time that they can also promote the glioma growth. But here what we've seen also that the astrocytes and the neurons can have the role. And also what they have observed is that actually tumor by their signaling could control also the neurons and how they send the impulses within the brain. So what we thought there, okay, with X-rays happens like this, but what we can do with the particle therapy? So is increased LAT going somehow to help us to destroy this kind of network? So our hypothesis was that the particles will form complex DNA damage, so less dependent on the ROS, because one of the postulates was that the ROS is being transformed through these microtubes and giving certain survival signals to the tumor cells. And basically this difficult to repair damage will happen, and the cells will disconnect and they will eventually die. So here we were using also, again, patient-derived cells from glioblastoma patient, and we were again comparing between proton, helium, carbon, and oxygen ions, the damage, and then we saw similarly what I've shown before, that there is the increase in the radiation-induced foci in their complexity and size as well as the number, and we saw that this correlates with the tumor microtubes density, as we call it here, TMs here. So more foci, higher LAT, less connectivity, and less tubes. So you can see here, for example, in this case, these are not intercellular, but intercolonial tubes. I show them because this is also something really interesting. So when you do your clonogenic assay with glioblastoma or maybe even pancreas tumors, just give a look. Maybe there are these tubes that could be somehow interesting for research and to be targeted and could play a role in the resistance. So in addition to try to kill the resistant tumors, one of our aspects is also to try to spare the normal tissues. And there our lab actually, our colleague Mahmoud Mustafa, that Walter also previously mentioned, since last 10 years have been working on radiation-induced lung fibrosis. So we made models. So in our lab, actually, this was like an effort of many people, and since 15 years it's going on, is that we were able to develop something that we call fibrosis index, and we were able also to see here the cytokine profile, for example, from the lungs that are from the radiation-induced lung fibrosis versus the control lungs that were completely different. And what was interesting here is that we have seen that this fibrosis index was much lower for the carbon ions versus photons. So when we speak about sparing the tissue, then it's, we have to come to flesh again. So this is, again, I'm coming back to brain because brain is mostly my focus. And this was one of our first in vivo experiments with the protons. And this was a single Bragg peak in the cortical area. And we were watching just like histopathological results here and seeing that indeed, respect to flash versus standard dose rate, we have the less DNA damage, we have less endothelial damage, and we have also less of neuroinflammation and dying cells using CD68 and HNGB1 as the markers. So next we said, okay, let's what happens with helium, because there was a big helium, big interest in our center for helium, as Sammy was mentioning before. So we said, okay, when we have already helium ions, let's try to flash them and make it and see what happens. And we had a similar results with the proton. So again, with the flash, we had the decrease in the damage, decrease in the inflammation, and we have preservation of endothelium. Then we did one step further. Let's see what is happening with the tumor control. This is a subcutaneous tumor here, A549. This is like a lung tumor. And we wanted just to see what happens if we are able to control the tumor. As you can see here, it is a very similar tumor volume after the standard dose rate and flash. So similarly what Walter has shown before for the entrance and carbon ions and also in the peak, so that tumor control is still there. So what we are still also using are 3D organoid models, because in vivo is very important and we are very interested in the tumor microenvironment, and we cannot use that many mice because there is something called dry air principle. So I know that all in vivo people know what is this, so to try to reduce animal experiments, so we are slowly switching to the organoids. So we were able to characterize induced pluripotent stem cells, brain organoids. These are from the human stem cells. And we were doing some flash experiments using carbon ions in them. This was in the same package of experiments, first experiments, what Walter was doing with the CHO cells. So this was like end of 2019, beginning of 2020. And we have seen there that in the organoids, we had also reduced DNA damage after flash in comparison to the conventional treatment. So after the brain organoids, we are also getting the patient-derived tumor organoids, again, mostly focusing on the brain tumors, as for example, meningioma, glioblastoma, and since recently we are starting establishing and working in collaboration with the Professor Josef Penninger from Vienna on blood vessel organoids. Next is the oxygen ions, because there was, again, as I said, the possibility to also use the oxygen ions and the flash dose rates. And PhD student Celine, so she's almost finishing her thesis, was measuring the oxygen consumption rate using the different LETs from electrons, proton, helium, carbon, and oxygen ions. And what she's seen here is that the oxygen consumption was very similar after standard dose rate or flash, but this is very fine difference that always, I think if you really carefully notice, or you can read her paper for more details, it would take too much time to speak about this in details, that the oxygen consumption rate is always a little bit less after the ultra high dose rate irradiation. And then we try to see the oxygen effect, oxygen flash, on the clonogenic cell survival, because previously we were able to show in vitro in helium that at 1% hypoxia, flash was able to spare the cells in clonogenic assay. Walter seen it with CHO cells, but here, for example, after using oxygen and the flash dose rates, we were not able to spare these cells anymore. And just to mention, so the LET is something around 100 kilo electron volts per micron. And we tested also oxygen flash for the in vivo, for the tumor efficacy, for the tumor killing efficacy. And we had here pancreatic tumor irradiation. This is a KPC model, very aggressive one. And we, as you can see here, is similar efficacy, very low efficacy in respect to control. So we are not able completely to control this tumor, far from that, but at least we have a similar effect of flash and standard dose rate for the tumor volume and for the survival. And this is just my final slide and a small outlook. Actually, I wanted to show that since recently we are starting to work with mini beams with our particles in collaboration actually with a group from Paris, from Yolanda Presado. And this is our first step in this German-French collaboration. And I hope that with KN we could even more promote this German-French collaboration and work together. And I wanted to mention that we have a grant and an open PhD position. So if you know someone who would be interested, you can just write me an email and let me know. And thank you, everyone, for your attention.
Thank you very much, Ivana. Do we have a question for Ivana at the moment? One burning question? Yes, we have one.
Just don't ask me about the flash and oxygen depletion hypothesis. Otherwise, it will take too long.
Thank you. Very interesting presentation. You consistently see reduced DNA damage with your flash dose rates.
Yes.
How do you kill better tumor cells without DNA damage? Is it structural integrity?
So this was in the healthy tissue. What I've seen, this is in the healthy tissue shown.
Okay. And have you ever measured the type of cell death, the pathway of cell death in tumor cells?
No.
Thank you.
Thank you for the question. We go to the next speaker now. Professor Angelica Facoetti, National Center for Oncological Hadrontherapy, that will speak about the CNAO lab.
Thank you very much. And I'm going to give an overview of our CNAO radiobiology unit. And in particular, I'm going to address the question from CIMAC and the organizer to go more into the practical information how to deal with the growing activities in radiobiology in a clinical facility. So I'm going to really have a very quick overview of why we need a radiation biology unit into a clinical facility, and mainly I'm going to justify my position there now, and why then I will go through the most important milestone that we had and we reached in our unit, and then I will focus, of course, on the research interest and collaboration that are ongoing. And finally, I will also show you some opportunities for the external users. So, okay, we have seen today plenty of these pictures or something like that. When we talk about radiation biology research, we always think about the cluster DNA damages. We know that carbon ions give very important damages to the DNA, and so that we have this increase in effectiveness, biological effectiveness. But what we have really to take care and what we have really to consider is that it's not just a matter of having a conversion factor. So we just don't have a tool that is more powerful in inducing an effect, a biological effects, but we have to take into consideration that we are inducing a totally different response in our cells. We know, and we are learning, that different pathways are activated. And that means that we have really to think about carbon ion radiation biology as something that we have still to discover. We have a lot of things that we still need to know. And just if you want to have a very clear idea of what is the background of our knowledge in radiation biology with the carbon ions and protons, if you go to have a look on PubMed and you search for radiation biology, conventional radiation biology, general radiation biology, and then you look at carbon ions or protons, I mean, it's unfair, the comparison, of course. We know we have small number of facilities, but this is also amazing because we have a lot of things that we need to know, and we have a very big opportunity to improve also the use of carbon ions, the clinical settings, to promote also treatments that can be tailored on the patients. And I always like to put this image because it's my feeling on what we know about radiation biology with carbon ions. This is what we know, and this is what probably we'll have to understand, we still have to understand. And I have to admit that more I'm dealing with radiation biology with carbon ions, more this part is increasing. I mean, it's getting really, really important. So what we have in CNAO, we started to build a radiobiology lab in 2009. At the moment, these pictures are from the situation right now. When we started, it was empty, not so crowded, let's say, and then it grew up in the meanwhile. So we can irradiate, of course, with the vertical beam and also with the horizontal beam, and with water phantoms, and also with solid phantoms for the biological support for the cells or for the tissues or whatever. So the first milestone that we had to achieve was in 2010, was before the treatment of the patients, because in Italy, our Ministry of Health, our system, asked us to prove that both the proton beams and the carbon ion beams were safe for the patients. And so we had some in vitro experiments with the proton and some in vitro and in vivo experiments with the carbon beams. No science. They were quite boring test experiments because we had just to prove that in very standard procedures and experiments, our beams were again safe and were comparable with the other clinical beams. And these experiments were also performed by external investigators, so some national radiobiological groups and also people from Japan came in CNAO to make this experiment. So this is the first paper that came out from our group, and actually was the characterization, the in vivo characterization of the carbon ion beams. So we made also a comparison of our beam, radiobiological features of our beams with the Japanese one and also with the GSI. And starting from that, then we were, let's say, allowed to have fun with our beams. Things went on not so quick because, you know, priority was for the patients, and radiobiology, the time for the radiobiology was not so important. But anyhow, in the meanwhile, we also thought to have an experimental room because the previous we used only the clinical setting. So now we have a fifth beam, a fifth line that is for the experiments, and that was performed together with INFN and CERN. And this is a particular room. I put it these diagrams because they are better than the pictures, because as also from it, the room is not so fancy because it's just, it's not a clinical one. So it's a multiple configuration room, meaning that we can adjust the configuration of the beam according to the experiments. Of course, for us as biologists, we just like the standard clinical configuration, so we use this one. But for the physics experiments that they need a lot of space and particular features of the beam, this can be modulated according to the experiment. At the moment, we can have protons and carbon ions, but very soon we are going to have helium also for our experiments. And the positive fact is that experiments can be installed for many days and even during the treatment of the patients. Of course, the beam is only one, so the experiments must be performed during the night and during the weekends, as for all the clinical facilities. But we have the advantage to start to settle up the experiment during the clinical setting. The third milestone was the third source. That was very few years ago, and so now we have this third source that allows us to have helium, lithium, oxygen, and iron. Of course, iron is not for clinical setting, but CNAO is an open facility also for experiments that are not addressing only biological and clinical scenario. Then the next milestone. These are very, very next because we are almost there. As I told you, we started with that lab and we are still working that lab, but it's going to be small because we are growing, and so now we are really at the final stages of having a new area for radiation biology, and this is the map. Actually, we are going to have two labs, and in particular, we are having an animal house, a very small animal house, and this is crucial, and this is something that we really think is needed, and our experience said us that is needed, because first reason, we have some radioprotection issues that make not so easy to bring the mice outside of our facility. And second reason is that we are working during the night and during the weekends. And using the animal facility that is near CNAO, that is from university, is not so easy during the nights and during the weekends. So in this way, we can be totally independent. Plus, we are going also to have an irradiator room with a photon irradiator, again, for the mice. Because, for example, at the moment in Pavia, we don't have any facility where we can irradiate mice with photons. So that is, for example, the reason why we have a project with the GSI addressing carbon ions plus photons. So let's say that in a few months, we will move in and having more space. Then, of course, we are going to play with the helium that is already there, and we are just waiting the green flag for making the characterization, then oxygen and the other beams. And not less important, CNAO is expanding also with the BNCT facility, as a BNCT facility. So BNCT radiobiology is, again, another very, very big chapter. And we are going to deal also with this topic, of course. So which are our research interests? The keywords are always the same. I mean, it's radioresistance, of course, of after irradiation. And in principle, we are using tumors, cell lines that are, let's say, that reflect the needs of the clinic. So then also another topic is the migratory invasivity capacity of the tumor cells after radiation is another topic, and in particular the ones, the migratory that is driven also by neurotrophic factors. Then we have combined treatment. This is the experiment that is going on with the GSI to see whether carbon ions and photons, how to couple the two treatments, because at the moment in clinic they are mainly using carbon ion first and then photons, but there are no evidences in radiation biology with animals that actually this is the true way to deliver the combined treatment. And of course, radiosensitizing agents is a very important topic as well. And okay, immunomodulatory, immunosuppressive properties, we have heard a lot during this afternoon, and we have some research going on that. And of course, we are also addressing some new in vitro models, in particular spheroids and organoids and bioscaffolds at the moment, just before we will be, let's say, free to use also animal as well. Then new ions and BNCT, of course. So these are some of the projects that are going on, and this is a long list, and the time make me not able to go into the detail of all of them, but just to tell you that we have many grants, mainly national grants, coming from also the clinical experience. So I really want to stress the fact that having a radiobiology unit inside a clinical facility makes things really easier as sharing the information with the clinicians and with the people taking part of the experiments. And then I would like also to take the occasion to introduce the Hadron Academy. We have started 3 years ago a collaboration with the University of Pavia for a PhD course. So it's an international PhD course called Hadron Academy, and it is a PhD course open to all, say, to different profiles. So for clinicians, for biologists, for physicists, I mean, any person that can be involved, and you know, it's typically multidisciplinary field. So this is a very important way for us to make the research. And these are our collaboration. We have just signed, we are here, so we have just signed a memorandum of understanding with the the University of Caen with CEMAC for the research and training in Hadrontherapy. We have a cooperation agreement with CNRS and Tumour for several types of research. And for example, Marie-Claire is one of these example of this cooperation agreement. And then we recently also have restarted this collaboration with Michael Story Group, and now he is in Mayo Clinic, and so they just came in CNAO for making the experiments. And then, of course, these are all the other groups, some national, some not national, that share the research with us. Because again, we cannot stay in our own facility and stay there and do the research by ourselves, because it's really important, especially in this field, to collaborate. This is just a graph saying that we are open also to external groups. So this, we had some stop due to COVID and to some maintenance, but we welcome a group for scientific collaboration in our lab. And I would also like to point out this opportunity. I left some flyer outside from the E3+ project. E3+ is an European project on research infrastructure, and it gives the possibility to have two types of accesses, the research one and the clinical one. The research one is addressed for researchers that are willing to use the beams for making research that is related to clinical application. So in this case, researchers can have beam time for free, and they can be also reimbursed for their stay in the place where the beam is located. That can be actually at the moment at CNAO or Heidelberg. And then we have the clinical access. The clinical access is a new modality in this framework, and it allows clinicians, so people dealing with the clinical activities, to come to one of the facility in Europe to gain experience, to see how a treatment is performed, how the patients are discussed, and, for example, also for evaluating eligibility. Patient is not needed in this type of access, so it's just an application for people that really want to learn and to understand how to perform hadrontherapy. This is the website, and in this case, you are reimbursed for the travel and the lodging. And this is the flyer that is outside. And I like to thank my small group. We are growing. I started that I was alone, and now we are, they are all females. It's I don't know why. Sometimes some males were there, but I don't know, they disappeared. I don't know why. Thank you.
Thank you very much. Thank you, Angelica. We are really running out of time, so I would say we can go directly, to the next speaker. We can have the discussion later on. So the next speaker is Professor Piero Fossati, Scientific Director and Director of the Carbon Program at MedAustron. And of course, he will speak about the MedAustron Lab.
And as you said, long list of titles, but no radiobiology, so apologies. This will be relatively short, and I will try to make up for the lack of radiobiology with some blah, 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 or let's say with some personal opinion of what is the important missing point between the radiobiology and the utilizability clinic. As you know, we have in MedAustron one experimental room, which is used for non-clinical activities, and non-clinical activities means medical physics, so we do test of materials, MR guidance, so what happens to the beam under the magnetic field, accelerator physicist, organ motion, but there is also, of course, biophysics and radiobiology. This is a sort of strange situation because MedAustron is one facility, but the patients are managed by MedAustron, and the non-clinical activities or preclinical activities managed by universities from Vienna. So They are our laboratories, but they are not our laboratories in a way. So it is a very large cooperation, and all universities from Austria can access and do their research there. And it is equipped like a treatment room. You have the same nozzle and same dose delivery system and the same patient positioning system, which of course is not so relevant if you want to do small animals or cell experiments, but this is more for doing organ motion with phantoms and things like that. The quality is exactly the same as in the treatment room, so same verification and same beams. And this is really equipped with a lot of labs, full of all the equipment you would need. So this centrifuge, things to do DNA sequencing, irradiator with X-ray, a small animal house. We can treat cells and we can treat mice. And we have treated mice with protons, and then the first mice with carbon will be treated right now. Okay, so my role there is to really interact with our biologists and try to avoid useless experiments. So the point is, of course, we love science. Science comes out of curiosity. You want to understand how things works, and this applies to science. So if you want to find out about embryology, the mystery of life, how the cell works, the inner structure of the atomic nucleus, this is science. But for us, who cares about how ILET damages a cell? It is not such an exciting topic like understanding how the brain develops and works. It's interesting only as long as we can translate it to patient treatment. Otherwise, we would do more interesting things. So it is basic science, but it must have a translatability and this means that we have to talk more. So the only real point of my short talk is that we have to interact better between doctors and radiation biology. This is a typical example. We know carbon ion is more immunogenic, induce a more immunogenic kind of cell death. ITP, high motility group box 1, chirotecony. This is exactly what we daily interact with our biologists and say, if you don't scale for cell killing, what's the value of these numbers? It's nothing, because you cannot compare the physical gray. You have to scale for cell. This is just level 1 interaction, and if you don't talk to your other biologists, they don't realize it is a problem. I mean, if you scale for cell killing, it becomes very arbitrary. If you take just 1 gray, it's a nice physical measure, so it's cleaner and nicer for the radiobiologist to do the RBE of high motility group box. If this is higher than the cell killing, then it makes sense. If it is lower, then you should say, oh, indeed, carbon ion is less immunogenic, because this is... we identified within this E3+, which, by the way, Angelica mentioned, but let me mention again, if you are interested to come, E3+ is paying for the travel and accommodation, and you can spend some time and see how patients are treated with carbon. It can be the patient that you have referred, but you don't have to refer the patient. You can also come. And normally, it's very hard for senior people in a department to do that, but if you have younger residents, we are very happy to have them for 2 days, 1 week, 2 weeks. I suggest at least 1 week, but it is not really a hard condition. Within HE3+, we said, what are the promising, innovative use of carbon ion? And we identified hypoxia and reoxygenation, synergistic effect of carbon and immunotherapy, lead-based optimization. I am not telling you anything of this because it has not much to do with the lab. You do it entirely with physics and patients. You don't really need a lot of experiments to prove that it's a good idea to put the highlight in the tumor and not in the organ at risk. But I will tell you a couple of things of this and how it is important to have more interaction between the doctor and the biologist. So, first of all, hypoxia. We heard very well from Professor Toma-Dasu. I don't see her. Okay. Anyway, that it's not true that with carbon ion, hypoxia is not a problem. It is a smaller problem, but it is still a problem. These are, of course, the data from Ando, and the slide come from Guma. So I'm always indebted to the Japanese colleague. Now, the real issue, again, is we are focusing on hypoxia, and I think we should focus more on reoxygenation. And let me spend a little bit of a time, because this is something that we know, but I have the feeling we don't really know it to this level. Otherwise, you would be so enthusiastic as I am. This is the German data coming from Dresden and Tübingen mainly. So Michael Bauern and Daniel Zips made a lot of experimentation and investigation, and they were focusing on oropharyngeal cancer with negative predictive factors, so bulky and mostly HPV negative tumor. At the beginning of the treatment, these patients all had bulky tumor with FDG uptake and with F-MISO uptake. So they were using F-MISO to measure hypoxia, and these patients were all hypoxic at the beginning. But the point is that after 2 weeks of radiochemotherapy, some patient was still hypoxic and some patient was not hypoxic anymore. So we don't care what is the start. We care what is the mid of the race. Again, here we have horse racing. You can go to the horse race and you can place a bet. And you have to place a bet before the start of the race. It's not fair to wait until mid of the race. But we can be unfair in our practice with patients. Why should we decide at day one how to treat the patient? It makes no sense. You should decide how to treat the patient when you are almost at the end of the race and you know who is going to win and who is going to lose. We normally get excited when we have a 10% difference with a predictive factor. Oh, if I have this signature, then it will be a bit better. This is the two local regional control curves, okay? Look, not if you are hypoxic at the beginning, but if you do not reoxygenate after 2 weeks. You hardly ever see in oncology this huge separation. Okay, in the exploration cohort, you may say it's overfitting, there will be regression to the mean. This is the validation cohort. If you achieve reoxygenation, almost everyone is controlled. And if you do not achieve reoxygenation, you may even question, oh, is it worthwhile to continue? And this is very, very significant. It's a huge effect. So my question is, should we really say, let's use high LET carbon focusing here for this patient hoping to salvage the situation that will go very badly, or shouldn't we rather say, let's try to avoid this situation, not be there, make sure they do all reoxygenate. And there is not a lot of research going into the direction of reoxygenation. Even the mathematical modeling we have heard before, this is showing how you account for the reoxygenation that occurs. So it is something like given from the gods. No, we must determine this reoxygenation. We must act in the very beginning to drive the reoxygenation. These are, again, data coming from Japan. If you don't treat the tumor in the animal, it will grow. And if you treat it with low LET, which was photons, or with carbon ion, you can modulate your dose in such a way that the growth delay is the same. So you rescale for efficacy. And if you rescale for efficacy, at day 1 and at day 2, the same dose achieving the same local control. But day 1 and day 2, you get better and faster reoxygenation with carbon. We don't have a lot of clinical data, but we have some wonderful clinical data coming again from HIMAC-QST. It's mostly the analysis of Dr. Inaniwa, and the interesting subset of patients is early stage lung cancer. They were treating early stage lung cancer with 9 fraction, 4 fraction, 2 fraction, 1 fraction. And they were extrapolating which dose should I give if I reduce. And the bad surprise when you go to 1 fraction, then the extrapolation didn't work. The dose was not enough, because with 1 fraction, you have no resensitization. Now, of course, it's not only reoxygenation, it's a more complex process, but reoxygenation is a big part of that. And reoxygenation 4 fraction is a completely different word as 1 fraction. So even a short time, 2 days is actually interesting. So it means that we could, instead of using all carbon, as Esther was saying, there are some tumors in which using all carbon is very difficult, because you need a large volume of healthy tissue that will be very toxic if you radiate, for example, locally advanced lung cancer or neck with bilateral radiation. But if you take a tumor, and this is one example, non-small cell lung cancer may be another example, bulky gynecological cancer may be another example. So you wouldn't really think about all carbon for this tumor, but you might think about carbon up front. How do you do the animal experiment? You need an animal that has an hypoxic tumor. So here we have a real problem about the model. For me, this is interesting in a tumor that at the moment of the diagnosis, the tumor must be bigger than a mouse. If the tumor is smaller than a mouse, then it's not so interesting. So we would need bigger animals. Otherwise, we don't have this tumor in which hypoxia plays a major role. So is it worthwhile to do investigation in mice? Yes and no. Is it worthwhile to do investigation in mice in which you induce hypoxia by clamping? No. Hypoxia is not only hypoxia because you don't have the oxygen that stabilizes the free radical. Hypoxia induces and selects an aggressive phenotype. If you do an artificial hypoxia, it's not the same. So I would say either do it properly with a big tumor in a big animal or go from a preclinical in vitro to patients. But it would be misleading to do mice experiments there. So take-home message is hypoxia or reoxygenation. I would very much love to see more focus on reoxygenation, and we should be very careful about animal models. It might be that the model we are using is completely inadequate. Second topic, synergy with immunotherapy. We heard from Dr. Frey, from Professor Frey, that abscopal effect is a bit of voodoo. Yes, but we like this voodoo. I remember the first near CNAO meeting in Italy, and all the colleagues from radiotherapy attending to this and showing the excellent result of Dr. Yamada and Dr. Kamada and Dr. Tsuji, and they thought it was voodoo, but it's now there. I mean, we want to have some really exceptional things. And this abscopal thing, patient is full of metastases, you treat only once together with immunotherapy, and they all disappear. This is what we really like. We would like to see it happening a bit more often. Normally, we try and it doesn't happen. That's the problem. Most of the time, we cannot reproduce, but it's not never. Sometimes you have the miracles. The point is, can we learn something from the biology so that we will be able to design a clinical trial that makes the miracle happen, not 1 out of 1,000 patients, but maybe 1 out of 100 or 1 out of 50 patients? And for me, we really have to consider the complexity of immunogenicity after carbon ion irradiation. So we have to consider time and space and drugs and homogeneity versus inhomogeneity. There are a lot of variables. Where do you put your irradiation according to the use of immune modulation? We heard it's very important, but we also heard, it's very important to see if you irradiate the lymph nodes or not. At the moment, we are trying to explore the possibility of increasing even further the immunogenicity of carbon ion using some drug that maybe by itself would not be so well tolerated, but is going to shift. We want to use radiation as an in situ vaccination, and to make this in situ vaccination even more effective, we could, for a short time, give at the time of irradiation a drug that drives the response to the radiation-induced damage. Also for this, we have a major problem with animal model. The standard abscopal, you treat one tumor, you don't treat the other, you do it together or without the immunomodulating, and then you count, can I control the tumor I've treated, can I control the other, can I control the metastasis? This is what happens with the tumor. You heard, again, from Professor Frey, it's relatively easy for the immune system to recognize and kill the cancer. Why doesn't it happen? It does happen. Most of the time it does happen, but then you don't know because it is not a patient and you don't see it. You only see those patients where it did not happen. So tumor has a lot of time to learn how to bribe the immune system, to corrupt the entire system, that the police are on the payroll of the tumor, that the judge is on the payroll of the tumor. It will not be attacked. But if you go for animal experiments, you skip completely the immune surveillance and editing. No surprise you can achieve a nice abscopal effect, but this will not. So for very big things like which drug, okay, but if you want to go to the fine-tuning, how to combine the best fractionation scheme in space and time, what's the value of an animal that has not undergone immune surveillance escape? So for the immunotherapy, we either go directly to the patient or we need to find an adequate model, and I have the last 10 seconds. there was this brilliant idea from Mike's story that the adequate model might be a spontaneous tumor, so treating pet companions. We should really not forget this. There are a lot of dogs with osteosarcoma. We don't want to do cruel research. We want to treat them as a patient. We could treat dogs for free, and we just ask to make an autopsy if they die, and then you can try all sorts of spatial fractionation. So those would be my point about how to bring together biology and clinics in a more coordinated way. Thank you so much.
We just finished the mice and rat facility. You know, if I come with the idea of dog, they will kill me. So you have to consider also this. But okay, so I would say there is no time for question now. We can go to the next speaker, that is Dr. Takashi Shimokawa from the National Institute for Quantum Science and Technology, that will give us an introduction about the NIRS lab. Takashi, the floor is yours.
Yeah. Thank you very much, Chair, and also I've expressed to my thanks to the organizing committee to inviting me for this nice workshop. So I'm Takashi Shimokawa from QST Japan, and I'm group leader for radiation research group and also the coordinator for biological experiment at HIMAC. So it's my pleasure to share our experience at the HIMAC. So just I want to skip this one. So actually our institute is very close to the Disneyland. So if you come to Japan, please step by our institute. So this one is the overview of our institute of the Chiba areas. So in this area, we have 2 electron accelerators and 3 cyclotrons and also 2 synchrotrons. These 2 synchrotrons is a HIMAC. So already Dr. Yamada explained about the clinical part. So for my presentation, I will focus on the basic experiment. This is the inside of the HIMAC. So of course, it's HIMAC is intended to the clinical treatment, but also we are intending to the basic research. Because 30 years ago, we don't have enough knowledge about the basic science of the particle beams. So therefore, in this facilities, we have a physics irradiation room and also biological irradiation room. And also, we opened this facilities for the internal and also external researchers to do any kind of experiment here. So here I show this operations schedule for the HIMAC, similar to the other facilities. In the night and the weekend, we did the basic experiment. But unfortunately, right now, this rising the electricity price, we stopped this weekend and holidays machine time, so we only have 2 nights per week for the basic experiment. So here's I show this collaborative research at the HIMAC. So we even started in the beginning of 1994. This collaborative research for not only internal, but also external researchers. So purpose is just 2, so it must be academic research, and also disclose on the information, either paper or conference presentation. Then this running cost is covered by the NIRS, our institute. Then this year's 3 medical projects and 36 biological projects and 32 physical projects was carried out. And then in this 36 biological project, 2/3 of the project was done by the outside researchers. It's external researchers. And also 12 projects were from the international researchers. So actually, so this is a distribution of this international users for our institute. So actually, this is a unique thing in Japan. So mostly, we are more focusing the national collaboration, but our institute is more focusing its international collaborations. So then this is a fact sheet of the research activities at the HIMAC. So this last decade, we faced several difficulties. For example, this COVID pandemic, and also this rising the excess cost is really hitting the machine time. Then, but still we're keeping this project numbers and also keeping outcomes. So we believe so this stable and continuous outcome is a result of the international collaboration. And also, we every year we publish this annual report of the research project at HIMAC. So you can see this kind of report in our institute's homepage. Unfortunately, the most part was writing in Japanese, but at least abstract was writing in English. So Chair is a little bit pushing me to shorten my presentations, to talking a little bit fast. But from here, I want to more focusing to our biological irradiation room, the details. So this is our biological irradiation room. So beam comes from this line. So we have only one horizontal beam lines. Here we have a binary filters and also collimator fields. So only we have horizontal beam, so we can irradiate a cell and animal like this way. And this irradiation field stage is very flat, just a flat stage. So whatever you want, you can do this on this stage. This is our basic supplied beams at the biological irradiation rooms. So from helium and ions, we can user can use for whatever you want. But as you know, for the biologist, so this radiation field setup is so difficult. We cannot do anything, also cannot understand. So therefore, our support team is set up all the radiation field just before experiment. So our radiation biologist or our biologist just bring own samples and put the stage, then can irradiate the exact dose and exact LETs. But if some group, they need special conditions like protons, xenons, whatever, so in that case, they must measure or prepare themselves this radiation field. But we can support this kind of experiment too. This one's I show this our standard beam conditions, 290 kilo mega electron volt per unit carbon ion beams. So usually we give this 10 centimeter diameter radiation field. This is a flat, just plus minus 5% dose. And in this field, you can use a monoenergic beam or 6-inch SOBP beams. So usually we use for this monoenergic beam for the small size samples like culture cells. Then the LET is 30 to 8 Kep per micrometer as recommendations. And also 6 centimeter SOBP beam we can use for large size samples, mainly the animal experiment, and LET is 40 to 70 Kep per micrometers. So in these conditions, so internal and external researchers was doing various types of the project. This kind of DNA repair or breeding or protection kinds, these kind basic radiation research. Also preclinical experiment also doing that here. So then I think I have a little time. Okay. So from here, so I was more focusing on explaining our radiation biological activities in QSTs. So here I show the QST, the organization, the chart. So right now, 4 radiation research group was working on this ion beam experiment. So among that, my group was belong to the Department of Accelerator and Medical Physics. This is in charge of the physical experiment in HIMAC. This I think the little bit strange structures, but it's good for us. So then, as our part of the mission of the department, we collaborate with Dr. Inanyo and my group doing the evaluation of the new irradiation method. For example, such as multi-ions or flash and grid, these kinds new treatment method is coming. Then we did evaluate these ones using the biological index. So for example, the RBE, the validations or safety testing or other experiments. So this is a good collaboration with the physical groups and also biological group. So then this is my group. So this actually small group. I believe this is the smallest group in today's presentations. Our group is mainly focused on the basic research for cancer treatment. But also we spend some effort to do some basic experiments for the other non-cancer treatment experiment, and also the radiation biological analysis of the particle beams, more basic experiments also do to spend some time for investigation. But it's a very small group. So therefore, we have some advantage to collaborate with experts on the individual field. Otherwise, we cannot do anything because it's only one researcher here. So then the last, this past 5 or 10 years, we really successful the collaboration. I explained several examples. The first one, this obscure effect, this is a hot topic and even today's. And we collaborate with Walter and Alex and the GSIs. We successfully published these papers and just skip the details. And also we collaborate with the French group, Dr. Prezard, and this is mainly focused on this mini-beam radiotherapies. And we evaluate the skin damage reduction using the mini-beams. It clearly shows mini-beam is really reducing the skin damage, even irradiate really high dose. Then for non-cancer treatment, we collaborate with Dr. Amino and Dr. Yoshioka, the Tokai universities. And this was actually it's a very long collaboration with Tokai University and our QSTs. We spent 25 years for the basic research, and we published several results to showing this radiation treatment can reducing this kind problem in the heart. And then last year we published this first clinical trial result here. So these kinds of collaboration we did, and of course not only the collaboration, we did spend more time for our own individual independent experiment. For example, the left side ones, we showed this regrowing tumors characterizations. So as you know, after the treatment, sometimes the cancer becomes malignant or radioresistant, so that's the regrowing tumor is really a problem. But we compare this regrowing cells after gamma ray radiations and also carbon ion irradiation, irradiated regrowing tumors. It clearly shows gamma ray repeated irradiations to causing malignancy of the tumors, but carbon ion irradiation is not changed so much. So this is really showing that carbon ion's advantage for the clinical aspect. And also here is another obscure effect of the publications. So we try to find a good way to induce the obscure effect, mainly to the irradiation method part. So then compare the 10 gray and 3 grays. So of course for local controls, 10 gray much better than 3 grays. But the point of view, the induction of obscure effect, 3 gray is much more inducing obscure effect. And on the other hand, 10 gray is inducing the phoenix rising effect. It stimulate non-irradiated tumors compared to controlled tumors. So these kind of projects also do doing our groups. So we have more information. You can see this from this homepage, or if you're interested in using our HIMAC, please contact me or this promotion office. So thank you very much.
Thank you very much, Takashi, for being in time. Perhaps there is just time for one question in case. Otherwise, okay, if there are no questions, we can move to the last speaker of the session. That is Dr. Hymac-QST. Yes, was correct? Yeah. Professor of Radiobiology, University of Cannes.
7 minutes. 7 minutes. So as you heard, I have only 7 minutes to present what's going on in Cannes, considering radiobiology. So actually in Cannes, so I tried to find out a common title for all the activities. There are 3 labs. So basically we are working on overcoming tumor resistance, integrating hadrontherapy and targeted therapy, and evaluating effects of healthy tissue. So covering both tumor and cancer cells. So as a starting point, I just also wanted to show you. So I'm actually representing these presentations in the behalf of Dr. Karine Rohan sitting here, François Chevalier sitting here, Samuel Valab, and also my part of the work. And it was not so easy to merge all these slides. So we start with the ABTU, the research that are done by Dr. Karine Rohan, my colleague. So what she is doing, that she has a 3 direction of research: clinical, preclinical, and in vitro research. So considering example of her clinical studies are, for example, the prediction of the appearance of late effects, using different models, particularly different, let's say, pathways involved in the appearance of development of side effects, late side effects, in this case, skin reactions. In the top, and then in the bottom, you can see that they are also trying with some of the medical doctors, trying to improve the STOP protocol. So as you see, there are an improvement of the STOP protocol for the glioma patients if they use a combination of the radiotherapy with the olaparib, for example. Considering the preclinical research that are, I see here, yes, preclinical research that are responsible, that Karine has responsibility for. So one of the main important interesting area is the comparing the delivering technique. In this case, comparing the scanned PBS, so scanned version, let's say, and with the scattered proton delivery system. Now, as you clearly see that there are some difference if you look at the PBS and DS. So actually there are only one, for example, proteins are in common, and they are quite different considering how we actually deliver the dose to the patient. So that should be very important and to be considered in maybe in our future research. We'll see after the discussion that we have later on. And also in the bottom, you can see that the prediction of the toxicity and also in combination with the DNA repair inhibitors that have been also done by our colleague Karine. So the last part of the work for in vitro study, she's working with the new target therapy. So I don't know why it's this happened. Oh, okay.
To check whether you are sleeping or not.
No, I'm quite okay. So as you see in the bottom left, so there are new drugs, new inhibitors that will be tested in relation to the radiation. So, for example, ATR and a new ATR and ATM inhibitor, CHEK1 inhibitor, and as well as the role of microbiota. She has both the animal model and also 3D skin model for studying this effect. So microbiota on the radiation effect. So jumping to the next lab, it's ICCT, sorry, responsible Samuel Valab, working on the mainly on the glioblastoma, so brain tumors. So as we know, as we heard today, so hypoxia is one of the characteristics of, for example, glioblastoma, and this could be an interesting and important issue considering the infiltrative ability as well as the resistance of this type of tumors. So considering these two issues, so there are several mechanisms. So one of the important issues that it's dealing at that lab is the number of the proportion of the cancer stem cells in that, and then another research area that are doing there is that how radiation, radiotherapy in general affect the normal brain tissues. So I will also very fastly go through what's going on there. So as you see here, no, you cannot see here, but if you look at the left the left figure up there, so you can see that this cells, glioblastoma cell line U251, have been irradiated, whether in hypoxic condition or non-hypoxic condition, by X-ray and carbon ion. The top two are the X-ray, and the black line, there are two lines there overlapping each other. So it's a survival of these cell-lined cells. Actually, the cancer stem cell has been isolated and then irradiated, and the survival was established. So you don't see any effect of hypoxia there, considering carbon ions. So this is an actually, I didn't show you the results of another cell line that it's responsive in relation to hypoxia or oxygen concentration and irradiation. But it seems that for particular cell lines or particular tumors, so carbon ion, although increase the cell killing effect, but the level of oxygen has no effect. So it means that should be something else. So this lab are very interested to understand what are the other factors that can affect the let's say survival in hypoxia or non-hypoxia. So one of the experiment that they have done, if you look at the X-ray comparing with the proton and photon, or if you look at the right figure, you can see that even 8 gray of radiation can actually stimulate those stem cells to grow more, while for the proton and for carbon ion, this number of the, let's say, tumoroids are quite low. Also, perhaps one of the important issues to be considered is X-ray perhaps stimulate the cancer cell proliferation, cancer stem cells proliferation, and that's why we do see this high number of tumoroids, but we don't see in proton and carbon ion. That is an interesting observation that are being evaluated there. So considering the effect of the particle radiation on the normal brain, so there are study going on, but the main conclusion of this study is that although the dose is maybe lower by the proton and carbon ions, but there is effect on the primary stem cells, primary neuronal stem cells to be considered. It's in the brain irradiation. And another project that is interesting that has also been touched here a little bit was effect of radiation on lymphopenia, and here the group has irradiated mouse model, and then they were looking at what type of lymphocytes or leukocytes in general been changed by the whether proton versus X-ray. So as you see in the top left, so by using X-ray, you really decrease the lymphopenia, decrease the number of the, let's say, leukocytes, most of them. But using proton, you don't see such a changes, such a huge changes, and it could be due to the volume that it's, for example, irradiated by the using the proton and also maybe other type of hadron. So it's the normal tissue volumes maybe is less. So the team area at Ganil dealing with the basically there are 3 axes of research. First of all, in how to overcome the radio resistance. Generally, most of us doing that, but they have very specific and up-to-date and new, maybe new inhibitors. I will show you a little bit faster. And then a project on the flash carbon. And traditionally, this lab was deeply involved in the bystander effect of radiation and bystander effect of the Hadrontherapy, which is quite interesting. So the system that are used in the ARIA laboratory are chondrosarcoma, glioblastoma, lung cancer, head and neck cancer model, and the targeted radiosynthesizers, PARP inhibitor, KRAS inhibitor, ATR and ATM inhibitors. So a combination of them with the different type of radiation, and as you see in the last line, so it's with different concentration of oxygen, so hypoxia or non-hypoxia, and also focusing on cancer cell biomarkers. So I think I will skip some of those because I already said about the FLASH, which is doing at the moment in ARIA laboratory, and also on the non-target effect, so bystander effect, but it's extremely interesting issues there, working using the carbon ion, comparing with the proton and X-ray. And I will go through what I am doing. The model that I am using to overcome for the radioresistant or cancer stem cells isolated from glioblastoma, rhabdomyosarcoma, pancreatic ductal cancer, colon cancer, and colon organoid model cancer. And for the healthy tissue, we are focusing on bone marrow and adipose-derived stem cells, survival and differentiation, and using also healthy colon organoid models will be used. I haven't used yet. The last slide is about a project that we started for some times ago, 5 years ago, a European project called Harmonic, and this Harmonic gives us opportunity to collect biosamples from the pediatric patients before the samples, before the treatment, after the treatment, and long time after the treatment, 1 year after. So we have been setting up this biobank in Stockholm University, but this will be moved to Caen. So in the place where we have, at the moment, ABTU, we will have the opportunity to have all the biobank there with the blood samples and saliva samples from pediatric patients, proton versus photon, just to understand, is there any biomarkers that could be indicative for the long-term effect of radiotherapy. We are very focused on second primary cancer, on chronic inflammation and chronic oxidative stress, which could lead to neuronal or cardiovascular disease or neurodegenerative effects. So I will finish here because we are a little bit hurried to start the discussion.